Good afternoon, everyone. I'm Debbie Ingalls. I'm the Director of CCOVI. Thank you for joining us today for another session in our CCOVI Lecture Series. This afternoon, I'm pleased to present Dr. Sudarsana Poojari, who is our Senior Grapevine Virologist here at CCOVI. So for a little bit of background on Sud, he earned his PhD in plant pathology from Washington State University in 2013, where his research was focused on biological and molecular aspects of graft-transmissible diseases in grapevine. Before joining CCOVI and after his PhD, Sud completed an NSERC postdoctoral fellowship at Agriculture and Agri-Food Canada at the Summerland Research and Development Centre in British Columbia. And his work there focused on understanding the epidemiology of grapevine virus diseases in British Columbia. So here at the Institute, Sud's research is focused on developing advanced molecular diagnostics for grapevine viruses and understanding grapevine virus vector interactions. We all know this is an important issue, not only here across Canada, but also for grape growing regions around the world. And Sud is a big contributor to knowledge going forward in the field. So today he'll talk to us about some of the latest research updates in understanding grapevine virus diseases. Thanks.
Thanks, Debbie. Before you all came here, we were just thinking about, some of the nicest places in this crappy weather. And one of the places we were thinking is, going to warm weather conditions, maybe Caribbean islands or, Hawaiian islands to take a dip in the sea. So I'm bringing this to, lighten you up, probably bring your attention to tell you that, in a drop of seawater, there are close to 10 million of viruses in it. When you go take a dip next time, remember that you're living in a sea of viruses. The point I'm making here is they're not all as bad as some of them we see on grapevine viruses. They're mostly the viruses that actually fed on the bacteria in the seawater and not necessarily cause any harm to humans. So don't worry if you want to splash some water around in the sea, that's fine. But the thing I'm making is probably these viruses evolved from the sea because that is considered as the most abundant source of viruses in the entire planet. So they gradually make their way into the plants and, of course, on the grapevine viruses. So today we are going to talk of some of them on plants. So there are many viruses, obviously on grapevines grapevine is susceptible to maybe 60 or 70 different virus species, different genomes. They have different capacity to infect, but not all of them are important, as I just mentioned before. So today we're going to probably focus on these four basic questions on how many viruses that are important to our grapevines, and what type of symptoms, will the symptoms indicate anything, and also how they spread, and what we know so far, some of the major virus diseases, and what type of options that we have in terms of management. So as I said, there are so many viruses. I'm not going to list all of them, but it is very important for us as an academic, as well as industry, as well as any students that are here in this hearing this lecture, to know what are the regulated viruses in Canada before we talk about anything of importance in terms of viticulture and enology. So CFIA, Canadian Food Inspection Agency, which is the regulatory body of the Canada in terms of quarantine, has listed these many viruses in the quarantine list to make sure we don't get these. So many of the times what we test here or we do on research, if we test for these viruses, we are obligated to report to CFIA. That's in a mandatory situation, and they will probably take some necessary action to sort out those situations, whether to control them or to minimize the effect of these viruses. So we're not going to talk about this, but we're going to talk about some of the viruses that are listed in red color here. So as some of you might know already, but there are 4 or 5 disease complexes that we really concerned because they do cause some kind of a damage in terms of economics are concerned. So first in the list, as you see, grapevine leafroll disease. We'll talk about that one a little bit, and then the grapevine red blotch disease. Sorry about that spelling mistakes there. And then the Rugose wood complex and Fanleaf degeneration complex. And we also talk about a little bit about this new emerging virus disease called grapevine Pinot Gris virus. So the first one, as you can see, this is, every time we talk about the disease in the grapevine, we talk about a word called complex, because some of these viruses are not characterized in terms of how specific each virus can cause a disease that will show symptoms, and that is the actual causative agents. That's why it's a complex, and it is always associated with this disease. Although we have characterized some of the viruses that are associated with the Leafroll disease as a grapevine Leafroll associated virus 1, 2, 3, and 4, but the whole disease complex itself has not been studied in detail. So why it is important? It does cost money. Do we have any data to show? We don't have any such economic data to show under cool climate conditions like what we have here in Canada. But in the United States, for example, they did economic studies that shows significant economic losses, especially for this Leafroll disease. For example, in California, the amount of losses that you can see is close to 29 to 226,000 US dollars per hectare. Of course, these surveys were mainly depend on what type of management practices that you are carrying in your vineyard and how much the disease incidence is there. Similarly, in the New York, the amount in US dollars anywhere between 25 to 40,000 US dollars because of the grapevine leafroll disease. Slightly different study in terms of how much revenue will decline based on the market value of the grape. That was done in Washington State. As you can see, in this particular study, they estimated 30% yield loss that is equal to 1% sugar reduction. That can cause up to 77% in revenue decline. So what does this tell us is over the time, if not immediately after the infection of the disease, you can see a gradual decrease in terms of production. And the economic losses, obviously, you can see in Leafroll disease as well as the red blotch disease. So we see those values, but we have to compare where those values are actually coming from. So as you can see here, the virus, the Leafroll disease affects both viticulturally as well as enology side as well. In the wine aspect, if you see graft incompatibility issues as well as the depend on the rootstock and the variety and the clone, it will have an impact on how successful your grafting material is. In terms of fruit quality, you can see lower soluble solid, especially sugars, that again increase the acidity, lower in anthocyanin concentration as well as the pH, which are the very critical components for the final product that is wine. In terms of final product that is wine, in the sensory evaluations, you can see it is when you make wine with the Leafroll infected versus healthy, you can see less color, less fruit aroma, more astringent. We have heard about these effects last CCOVI lecture with Pat Bowen talking about the effect of Leafroll 3 infection on grapefruit cultivars in the Carnatic Valley. So I wanted to show this animation here, which is recently published by Peroni et al., which actually describes you exactly what is happening in the virus-infected wine, especially on the red-fruited cultivar. On the left side, you have Leafroll 3 infected, and the right side, Leafroll 3 free. As you can see here in the graphic, what is happening here in the leaf is the decrease in the photosynthesis that is because it is a result of downregulation of photosynthesis genes, and then upregulation of defense-related or stress-related genes, as well as anthocyanin in symptomatic leaves. The main reason for this is these grapevine leaf roll-associated viruses are phloem limited. They're only which means they're only restricted to the phloem tissue. So what it does is when they replicate and when they propagate more, when they increase their viral population in those phloem tissue, they block it. They block the source sink mechanism. So the photosynthesis that happens to produce the sugar, which is normally transferred onto the berries to get more sugars, and then when the sugar accumulation increases, we know by research that the anthocyanin pathway will be triggered, and then during liberation, the color change happens in the berries. But if the plant is infected with the leaf roll virus, the sugar accumulation actually happens in the leaves, and then plant probably thinks that it is going under senescence or stress conditions, so the anthocyanin pathway triggers, and that's why you see this red or blotchy color formation. And with that, the level of photosynthesis will decreases, and consequently you see uneven ripening of the berries. As you can see, even with the research that shows that downregulation of sugar transport genes, as well as the metabolism of cell signaling genes, including the anthocyanin biosynthesis pathway. So we have done a study looking at the anthocyanin pathway, and we see the upregulation of anthocyanin gene in Leafroll 3 infected leaves that contributes to these symptoms in red-fruited cultivars that we published in 2010. So when we talk about the leaf roll complex, we just mentioned that there are different type of different species, starting from leaf roll 1 to leaf roll 10, different viruses. So among them, if you look into different viticultural areas in the world, which one is the most prevalent? So I put together a few slides that derive from different studies of surveys that have been done in different parts of the world. For example, this one in South Africa, as you can see, among the leaf rolls from here to here, that did 4 different leaf roll species. So you see the leaf roll 3 incidence is high all the time. So we talk about these viruses, GVA, GVE, GVF, and RSPAV a little later. But as far as the leaf roll 3 concerned, you see that the leaf roll 3 always dominates. Similarly, a study in California, again done in 2010, looking into 4 different major grape growing regions. As you can see, the leaf roll 3, which is in black over here, the dominance of leaf roll 3 infection. Similar study in New York by Mark Fuchs et al. And they also studied as, don't worry about the small letters here, but the bottom line here, which is underlined in red here, is the leaf roll 3 infection is more in red and white fruited cultivars in the Finger Lake regions. So with this information, we started a comprehensive survey in British Columbia a few years back, which we did with a sample size of close to 3,000 composite sample that represents around 15,000 individual wines from different parts of the British Columbia, wine growing regions of British Columbia. As mentioned in this graph here, we divided into 4 regions to simplify how the regional spread of the virus can be seen. As you can see, North Okanagan to Central, South Okanagan, and the Similkameen Valley. You can see we tested for 4 different species of leaf rolls, in all the regions, it is very clear that the Leafroll 3 is the most widespread. When you look into different regions, the South Okanagan, of course, the highest in terms of wine grape production, have the Leafroll infection with the high incidence. So this kind of information gives us an option to see we can design some management strategies based on the region, for example. So one of the interesting things that we did along with studying the virus incidence in this particular study, we also looked into the vectors of the virus. We'll talk about the vectors in a little bit, but we know that the grapevine leafroll viruses are transmitted by either mealybugs or soft-scale insects. So we surveyed the insect populations as well in this region, and interestingly, we found that in the North Okanagan and the Central Okanagan, the mealybug population is considerably less when you compare to the South Okanagan, which probably explains the high incidence of virus diseases. And then in the Similkameen Valley, which is a bit separated from the Okanagan Valley, if you know the geography of British Columbia, we found no evidence of mealybugs, but we do find a soft-scale insect population. That also gives us critical information in terms of how you proceed with the management of the vector to stop the secondary spread of this virus disease. So when you talk about the transmission of these virus diseases, because of the clonal propagation of these grapevines that we do, the primary transmission of all the pathogens happens through grafting material, either it is through a rootstock that is infected, or a cyan material that is infected, or if you are going for a bone-rooted, if you are propagating from a virus-infected mother plant, that's the main source of the virus infection throughout the world. When you look at the secondary transmission, especially for the Leafroll disease, there are many species of mealybugs or soft-scale insects that has been reported that can transmit Leafroll disease 1, Leafroll-associated virus 1, 3, and 4, but not for the 2, because there's no insect so far that's known to be capable of transmitting grapevine Leafroll-associated virus 2. But we'll talk about the mealybugs here because through our studies in Okanagan, in British Columbia, as well as preliminary studies here, we know that in Canada we have only one species of mealybug, that is dare mealybug. So, there's something cut off here. That looks good there. So, if you look at the grape mealybug generation cycle, how the population dynamics changes, or the life cycle of grape mealybug, it starts early March and April, especially when the temperatures of the ground temperatures rises above 8 to 10 degrees centigrade, and it is consistent probably for a period of 5 to 6 days. That's what our observation based on PC conditions, but I'm sure it will be same here. So the grape mealybugs overwinter as eggs, and they under the bark in conditions like this, where this cottony, puffy moss, and they overwinter their eggs, and when the temperature reaches above 5 to 6 degrees or close to 8 degrees, they hatch, and I'll show you how they look like when they hatch. So that's how they look like. Those we call as the first generation crawlers. The first generation crawlers can be seen from even until June because of the generation gap, how the environmental condition favors them, especially relative humidity and temperature. They can be seen until May and June. When the first generation nymphs mature, mate, and then return to the woody regions of the vine to lay eggs inside. So what we call these ones as the second generation crawlers, they are big. In terms of different stages of life cycle, this first generation and second generation crawlers are very critical because those are the most efficient in transmitting the viruses, especially Leafroll viruses. So this is the adult mealybug. As you can see, the size is 0.025 to 0.5 inch. The second generation crawlers usually emerges in July and September and make their way into shoots and grape clusters. They will mature quickly before they return to the woody portions of the vine to lay egg in the winter, where they overwinter. So one of the indicators that we see in the field conditions is you observe for the presence of ants, because there seems to be a symbiotic relationship between the mealbugs and the ant species, because ants harbor them, protect them, so that they can get a honeydew that is generated from these mealybugs. The mealybugs excrete the honeydew, and ants use those honeydew. So for that purpose, they protect them, and probably in that way, they're probably carrying some of these crawlers on their bodies and probably transmitting to the next vine. But as you can see, these crawlers are very active. They can probably walk from vine to vine in a very quick time. So that's the size of a crawler, just another picture taken with the microscope. So switching gears here, we go to the next virus, the grapevine red blotch virus. So see, the grapevine red blotch virus is quite new to grapevine virology community in terms of how far we know this particular virus. Although there are some reports as early as 2007 from Calvi et al. In California, they observed in one of their experimental vineyards the symptoms that are close to Leafroll disease, which is again red purplish color blotches. But I think it's been ignored, thinking that it is a Leafroll disease and not looking into different directions. But they reported as a red leaf disease, or some people call it as some kind of phytoplasma disease, but not until 2012 when we have access to a new detection methods called next generation sequencing. So the Trench et al. In 2012, they used this next generation sequencing to see if we can characterize that kind of symptoms. If it is a virus disease, and that's when they call it a grapevine red blotch associated virus. But after that particular event, people started to report from different parts, as you can see, from Washington State, California, as well as in California, the reports from table grapes. That means the virus was there. We were just thinking that it is something else, or as we're probably mostly confusing with the Leafroll disease. And from there on, there have been reports from different wine-growing states in not only in the U.S., but there are reports from Korea, Switzerland, even China recently. And in 2016, we reported from Ontario as well as British Columbia. And one particular highlighting aspect is that alfalfa tree hopper has been reported as a possible potential vector for this grapevine red blotch virus. And then going more into the details, Barbara et al. In 2017 also reported that the red blotch virus affects the berry ripening, very similar to what the effects of the grapevine leaf roll does. And then in 2018, Eaves et al. From Mark Fuchs' group in Cornell, they fulfilled the coach postulates, proving that this is the sole organism that causes this particular disease. So that's why we removed that associated name from the virus, virus title, so now we call it the grapevine red blotch virus. And then Patterson et al. In 2018 also identified red blotch virus in grapevine vitis repository germplasm in the US. What it does mean, that means except vitis species, what it does means is the virus was there for a long time. We were not able to detect it, especially the cultivars that are wild, the virus is present in them, which means that there may be wild species that is out there acting as reservoirs for this particular virus, and probably due to the change in the climatic conditions or viticulture practices, it is moving from wild varieties to the cultivated varieties like Vitis vinifera. The symptoms are very similar to Leafroll, but in some varieties we don't see the rolling back of the leaves. That's probably why they call it grapevine red blotch virus, because of the blotchy symptoms. But that being said, we study more, we learn more. We also see the rolling back of the leaves with this particular virus infections in some cultivars. And in the red-fruited cultivars, it's very obvious. You can see these kind of symptoms, this blotchy pattern. But in white cultivars, it's a bit confusing. It can be confused with many factors like especially nutrient deficiency or mineral deficiency. As you can see in this picture, the Chardonnay with the confirmed test for the grapevine red blotch virus. This kind of yellowing and also sometimes you can see the necrosis at the edges. That's very peculiar symptoms that we observe for the grapevine red blotch virus. So what we know about the vector, as I mentioned, the alfalfa tree hopper has been reported as the potential vector, but the experimental are conducted under the laboratory conditions. But still, there's very active research going on, not only in the United States as well as here. We've been trying to find whether in Ontario conditions, whether we have a different species of that. On that particular note, there was a report published by Mark Fuchs et al. In 2016, where they were studying if they can find the grapevine red blotch virus in different species of tree hoppers that belongs to Membraceae family, as well as some of the leaf hoppers that belong to Cicadella family. And they found some of these species that are mentioned here, the virus is present in those species, but that doesn't mean they can be vector of this virus. What we have to do, probably more research is needed at this stage to see they not only they carry the virus, but they have the ability to transmit from vine to vine. By the way, this is how the alfalfa tree hopper looks like. But through our preliminary studies that we did in Summerland Research and Development Center as a part of my postdoctoral program, as well as preliminary studies here, we found the tree hopper species that we have here predominantly is not alfalfa tree hopper, but the buffalo tree hopper. So as you can see, some of the characteristics, very bright green color. They have a triangular head, as you can see here. Very easy to identify because it's big. In terms for the insect, it's 6 to 8 millimeters in long and transparent wings. And it is not frequently found in grapevines, especially the commercial vineyards. But we always found the very low populations where there is a riparian habitat close to the commercial vineyard or some kind of water source or a canal running through. That's kind of places where we found this kind of species. But we're still working on trying to see we can prove the ability of this particular species as a potential vector or not. In terms of how to see, how to identify this kind of tree hopper infestation in the vineyards, as you can see in these pictures circled here, the end shoots, either you can see the necrotic brittling inside, as well as it is sometimes it is protruding outside. Even in the lignified canes, you can see this kind of girdling. That is the indication of tree hopper damage. So going quickly to the next complex, Rugose wood complex, which in terms of the Rugose, the meaning is wrinkled or corrugated. It is known for typical modification of grooves or ridges on the woody cylinder, especially when you talk about the grafting material, scion or rootstock or both. You have this kind of modifications, and these modifications, they're different on different rootstock or cultivar combinations, known to cause again synergistic interactions with other viruses and viroids. And the viruses that are associated with this, I have in the next slide, but most of this Rugose complex, the viruses that are known to cause this disease, are very latent or symptomless on European as well as American rootstock species, as well as the hybrid, but they do show symptoms on Vitis rupestris. In terms of viruses, even it is a complex, there is again 4 different type of disorders that are reported on this, which is called Rupestris sempitting, covert stem grooving, LN33 stem grooving, as well as the corky paw. And as you can see the symptoms here, the Rupestris sempitting is spitting on the stem. You can see here that is mostly caused by Grapevine Rupestris sempitting-associated viruses. And as I mentioned, most of the time it is very difficult to identify any plant, any vine that is free of the Grapevine sempitting-associated virus. But it's Grapevine sempitting-associated virus, or simply GRSPV, with a single infection, the sole infection, it cannot produce any symptoms on its own on any of these Vitis rootstocks. But when it is combined with the other viruses that belong to this complex here, the grapevine virus A or B or D or E, then probably you can see a little to moderate kind of symptoms, but not at the level of significant economic losses. So one of the papers that's just published quite recently by Pantello et al. Is described here. As you can see, one of the good things happens with the virus. This one, Grapevine sempitting-associated virus, again here. When you give a plant a little bit of stress, here it is indicated as a drought stress here, plant tends to perform, or vine tend to perform better when it has only infection of Grapevine sempitting-associated virus. As you can see, the photosynthesis and CO2 fixation going up, the defense-related genes going down, and in the paper they also described a lot of microRNA interactions that are activated for this kind of response, as well as the increase in the stromatic conductivity, as you can see here. But we need to be careful. If the Rupestris sempitting-associated virus is present in combination with other viruses or other viroids, we might not see this kind of effect. Probably it's a reverse. So about the Grapevine Fanleaf disease, it's another disease complex that is transmitted by a soil-borne nematode, that is dragon nematode, which is Zyphonema index. So far in Canada, Zyphonema index is not reported. So the symptoms, particularly you see, as the name indicates, Grapevine fanleaf, the leaves become feathery or fanleaf-like symptoms here. Chlorosis, patchy chlorosis in the vineyard is probably a good indication to suspect for this kind of virus. As well as you can see the virus slows the growth of the plants, so you can see stunted plants, irregular fruit clusters, as well as the short internodes. And sometimes, when you have symptoms like this, it can be confused with the herbicidal damage. I have another picture of the plant that is tested positive for the Fanleaf virus. As you can see, the leaf symptoms. But of the particular interest, you can see here the short and internodes. That's one of the things that we have to focus when you're looking for this particular virus. And also, when we talk about the symptoms, what they tell, what they do not tell, for the Grapevine Fanleaf virus, especially on the viniferous, you have to see these kind of symptoms in the early spring, not as what we use for grapevine red blotch or leafroll viruses, we started to see the symptoms at the fall. But for the Fanleaf virus, it is early spring that you see the symptoms. And most of the time, these kind of symptoms for the Fanleaf virus, they do gradually disappear. You don't see these kind of symptoms in the fall. So it is very important if you are looking for the signs or symptoms, you see the grapevine Fanleaf virus in spring. So the Pinot Gris virus. So it's been recently reported, probably last 2-3 years. It belongs to a completely different genus in the family Beta flexiviridae, and the genus is trichovirus. It's been reported from different countries so far: USA, China, Canada, and a lot of European countries, including France, Italy, and as well as Australia. So the GPGV known to occur both red and white fruited as well as the table grape. Mostly it is reported as the latent infection, but another peculiar aspect of this virus is it's been characterized with 2 different strains, one that causes symptoms, one that doesn't show any symptoms, we call asymptomatic. And also there are preliminary reports that says that the irinium mite or blister mite is possible vector for this virus, but there's still a lot of work to be done. On the other side, there are also the scary side of this. These alternative hosts have been reported for this particular virus, which is the couple of them that I mentioned here, lamb's quarters and white campion. These are obviously they are very widespread in Canada, and that's something that we need to take note of it if this virus is present in Canada on a large scale. So the symptomatic strain shows very peculiar symptoms. Again, can be get confused with herbicidal drift. As you can see, the leaves are mottling, and sometimes you can see chlorosis on the leaves. Similar to Fanleaf virus, you can see shortened internodes. The growth is stunted, but you can probably one of the viruses that we can clearly identify based on the symptoms, if it is a symptomatic strain. That's the grapevine Pinot Gris virus. So with that, I'm going to go and look to a different aspect here, the grapevine virus diagnostics, how critical they are, why they are important. As you can see, there are 4 different types of diagnostics here for the grapevine virus diseases. And these all 4 types of diagnostic methods are used in the clean plant programs, which is a critical part of bringing management aspects of this virus disease. One is the symptom-based, as you can see, based on the symptoms, but they can be non-specific and inaccurate. One of the things that we also study here is some of these virus strains, especially Leafroll virus 2, we do have evidence of asymptomatic strains. They are there, but they are in the latent conditions. So in that cases, there is a possibility to miss even in the red-fruited or white-fruited vinifera's. And also in the hybrid cultivars, most of these viruses, they don't show symptoms, so they stay as latent. But those hybrid cultivars may act as reservoirs for the transmission, for the secondary transmission. And then biological indexing. Biological indexing is best example is the grafting. If you have a rootstock that is infected, you graft it with a healthy scion, the virus transmits to the sand. So to characterize some of these viruses, we can also use some of these solanaceous plants, for example, as a biological indexing or an indicator plant through mechanical transmission. But again, it is labor intensive as well as time consuming. If you are just talking about the grapevines, the graftings, it might take a couple of years to see these kind of symptoms, as shown in this picture here. The serological ones, which is protein-based detection methods, ELISA is an example. They are very specific, and again, we have so many different type of viruses as well as their strains. But the antibodies that required to do this ELISA test, not all antibodies are available for all type of viruses. And again, when you compare to the next one, which is polymerase chain reaction or PCR, these are less specific in terms of their sensitivity. So molecular diagnostics include PCR, quantitative PCR. Another version of quantitative PCR is droplet digital PCR, as well as the next generation sequencing. So this is where now that all the clean plan program, the diagnostics are going forward. They're very highly specific, and they're post-PCR processes, but you can also have the ability to multiplex. That means you can detect more than one virus in a single reaction, and also you can quantify the virus. In overall, the bottom line here is the speed, sensitivity, as well as specificity, how robustness you can use, or how cost-effective it can be. Those are the things that can determine the clean plan programs here. So I just wanted to go through briefly about the clean plan program, how it operates in terms of this is again. A US model from Cornell University. So how it works when they have any imported or domestic planting material that has to be cleaned for the virus. As you can see here, when they found the virus in any plant, so the first thing that it will go to a micro tissue culture where they can eliminate the virus and then regenerate to the tissue culture. So it could take, as you can see here, it could take a couple of years to get to this stage. But once they got into this stage, this will go to the nursery mother blocks or foundation plant services in California. They maintain these plants and then they propagate it, which obviously takes 3 to 4 years, which then goes to the nursery blocks here before they propagate in the production blocks, further going to the vineyards. So to put it in a simplified way, in a classical as well as the next generation sequencing based methods. So I divided here these two parts here. As you can see, the classical clean plant program, when you have a planting material that has to be cleaned for the virus, if it is first they do the test for 30 plus different viruses through ELISA and PCR methods. If they're found positive, it goes through the virus elimination process, which is meristem tissue culture, and then they go to the biological indexing, as I mentioned here, which again takes time to confirm some of these pathogens, and then will be retested again for 30 odd pathogens, which obviously take days to week before it goes to the next stage where for the propagation of budwood and cuttings, where they can be mist propagated to get more numbers, and then it will go to the final full production, which again takes to 3 to 4 years. So the overall process here, we're talking about 7 years or so. So when they found the negative, but it can go to directly, propagating stage, but it will still take 3 to 4 years. But the advantage of next generation sequencing here, which is being adapted by some of these, clean plant programs around the world, is when you test it, which takes only days to week, it can directly go to, if it is positive, it can directly go to the virus elimination. If it is negative, you're directly going to the propagation stage. So that's the advantage of next generation sequencing. It is high throughput analysis, and there is no unambiguous results and no reflective testing. So what you're getting is a comprehensive report of what viruses or virus-like agents in this particular plant. Although there are some obligations in terms of what you found, what you don't want to find in that, some of the new things that you might find that needs more clarification in terms of how biologically active those pathogens are. But nevertheless, the next generation sequencing will give you, or will give you a comprehensive approach to test the grapevines and to know the status of the plant. So some of the options briefly in terms of management. Virus-free material is very critical, and the reliable laboratory test for the virus diseases in existing as well as to test the status of virus in dormant wood, or before using for propagation of the new vines, and not compromising on the quality of the planting material is very critical. And some of the reports, especially Ricketts et al. In 2015, have reported that the roguing of the vines is very important, especially if in the infection rate in terms of Leafroll disease. If it is less than 20%, roguing is recommended. If it is more than 20%, they recommend to replace the vineyard. Again, you have to consider how much the insect pressure in your vineyard is. The prevention of secondary spread by vectors is also very critical. You have to closely monitor the vineyards for the mealybugs or scale insect infestations, especially if it is a leafroll disease. And then also applying these pesticides to control these particular mealybugs or scale insects. The timing of the pesticides are also very important, and also studying the population dynamics in this particular region where this problem of the grape mealybugs or soft scales is very important to understand these timings. So just before closing, we need clean plants, but it's only the first step. The reason I tell you, this is the picture that I taken in the Okanagan Valley last year, and I'm sure you see half of the vineyard is already gone, and there is even the other half is not there. All these red patchy regions that you're seeing is loaded with the Leafroll disease. So when you have clean plants, we have to start clean and also stay clean. The reason I tell you this is a two-year-old plant in the same region, as you can see already showing the symptoms. So start clean and stay clean programs are very important in this case. So some more information, please refer to this. And I also thank all the collaborators, both from Summerland Research and Development Center as well as here in Ontario, and all the students and technicians. And that's all I have.
We've got time for questions from the audience. So anybody want to step up and ask the first question? Okay, I'll start us off. Good. When you were presenting the options for a clean plant program, you were talking about moving towards next generation sequencing technology to reduce the time required to get clean plants through the program. Is anybody using NGS yet for that purpose, like anywhere around the world? I know in Canada they're looking towards that, but there's some limits in terms of international agreement of technologies. Can you just expand on that?
Yeah, I mean, I'm talking literally on the US-based model. US is already moving ahead with this. Although because Canada with this European Union and we have to maintain those standards in terms of the plant protection regulations, but US is already there. They've been using in their domestic certification. But the international certification, I think that's where the concerns are right now, because there are different rules with the different countries, different pest regulations for different countries. That's where the actual problem, but they are trying to come with a common protocol to see that issue is addressed, because you're saving close to 2 years' time with one test. So, but US is in domestic certification; they're already doing it.
Any questions?
So maybe this is not your area, but if I can buy these clean plants and I have a site prepared, but I look out across the road and I can see symptoms of what appears to be red blotch or the leafroll in my neighbor's vineyard, how is that going to be addressed?
So that's one of the questions many people ask. It has to be a comprehensive approach. It has to be, dealt with, common goals. You cannot control this virus disease, which have insect vectors as a as a secondary source of infection that can either fly away or come from block to block. So if your neighbor is not controlling any insect vectors and they have the virus infection, it's very difficult to control. Even if you put the new clean plants, it's going to come. Of course, it depends on many factors, whether you are moving machinery from that block to here, or, the wind is blowing in the direction at a particular time where the crawlers, especially you look into different aspects of insects, for example, their crawler stage is more efficient in terms of transmission. Not only that, because they're very small, they can even be blown away with a strong winds like yesterday we got. So those kind of aspects need to be addressed, and it has to be dealt with a comprehensive approach.
Industry-wide. So we do have some representatives here today of the Canadian Grapevine Certification Network, and as an industry across Canada, they're working towards this as a common goal. Are we there yet? I'd say no, we're not there yet, but hopefully, as we continue to tackle these issues together, we come up with, common protocols and strategies that get implemented by everybody to help manage this. I know in Ontario, there's been a big investment in additional virus research to help us better understand the insect vectors transmission rates going forward. They're a little bit further ahead of us on that in British Columbia already, so we're drawing a lot of useful information from that. Those same studies are being carried out in Nova Scotia, and will likely be continuing in Quebec as well. I don't know, Jill or Matthias, if either of you wanted to add anything to that?
You pretty much covered it, Deb. I can't really speak to how the virus would be transmitted through the vectors, but if you're starting clean, it's better than starting dirty. If you're going to be planting grapevines. And as Deb said, we're in the process right now of creating those protocols, and we should have, we're running two systems right now, an interim protocol, which should have material available through it in about 1 to 2 years, depending on if you're looking at green vines or if you're looking at dormant vines. And then the long-term certification, which is going to be a lot more of what Sid talked about, where we're going through and testing vines and multiplying them out through G1, G2 mother blocks, about 7 years.
Any other questions? Okay, well, with that, Sid, thank you very much for an informative talk all the way around. Please join me in thanking you. And just an announcement, next week we've got a special lecture presented by Federico Tondini. He's a technical advisor for CellarTech, who's going to talk about yeast selection for cider production. Cider is on a growing boom here in the region, and lots of technical support from companies like CellarTech. So please join us either in person or online March 4th at 2 p.m. Thank you.